Okay, so this video is going to focus on the inner ear. And the inner ear, you can see how it is positioned in the model we went over for the external and middle ear. It has a bony pattern here, a bony shell basically, that's called the bony labyrinth. And it has three sections. It has the cochlea, it has the vestibule, and it has the semicircular canals. Now, it's attached to the middle ear again by the stapes. The last of the three ear bones is going to plunge in and out of a hole that matches the shape of the bottom of the stapes called the oval window. And it's going to take these mechanical plunging, kind of like a piston motions, and it's going to generate waves. Because inside this bony labyrinth, there's a layer of perilymph, and then there's a membranous, or like a water bag, filled with endolymph. Perilymph on the outside, endolymph on the inside. So this is where it is lying inside that petrous region of the temporal bone. And now I'm going to show you this model, which is this in the same position, just made really big. So I'm going to move this model now and be able to show you some of these structures. Again, this part is the cochlea. It's used for hearing. This middle section is the vestibule. It is for static equilibrium, and these three hoops are called the semicircular canals, and they are for dynamic equilibrium. So let's take a look and see the windows. So where that stapes would be, I'm going to turn this. Do you see this yellow structure right there on the bottom? That is the oval window. That's where the stapes will be plunging in and out. And if I turn this slightly, you see another little yellow hole on the bottom. That is the round window. So the energy for hearing, the little stapes, is going to plunge in and out of this window. And then this is attached here. This is the cochlea. And the cochlea has three chambers to it. So if I open this up, you should see a repeating three-chamber pattern. So this is really tiny. So let's look at one of these big ones over here. So this one is pink, this one is blue, and this one is green. So this is one section of this tube, okay? The pink section, the upper section, is called the scala vestibuli, or vestibular duct. And the divider between it and the middle chamber is the vestibular membrane. Okay, this is called the scala media, or cochlear duct. And this bottom one is called the scala tympani, or tympanic duct. Okay, so this is a small version, and it winds around and around. I'm going to walk over and show you a large version of this cochlear slice of just one of these sections. So let's walk over here, and I have it leaning against the board. Here we go. So now we can make out these structures much easier. So up here, this upper chamber, this is going to be the scala vestibuli, or vestibular duct, filled with perilymph. This is part of my membranous labyrinth. This part is called the vestibular membrane. Okay, this is my cochlear duct, or scala media. It is filled with endolymph. And then this bottom chamber here is called the scala tympani, or tympanic duct. Now this membrane is the basilar membrane. And then this right here is the spiral organ, or organ of Corti. It is made up of the tectorial membrane, which is the slab of jello-like material here. And then here are the receptor cells. They are hair cells. There are outer hair cells and inner hair cells. Okay, so how does this work? The stapes pushes fluid in and out through the oval window, which sets up waves up here in the perilymph. And at different lengths and diameters of the cochlea, those waves will pass through and vibrate the basilar membrane. When the basilar membrane vibrates at that particular frequency, then the hair cells will vibrate and bump up against this slab of jello known as the tectorial membrane. That bends the hair cells, will open up ion channels, and cause action potentials to be generated in these particular cells. So this is the dendritic receptor ending. Okay, we follow this cell. This is, again, the spiral organ. So here goes the dendrites. Here they go over here. And now we can see these round structures with dark circles. These are the cell bodies of these neurons with the nuclei. So this collection of neuron cell bodies would be a ganglion. And since these are the ones that are attached to the spiral organ, this is the spiral ganglion. And then these axons will leave these neuron cell bodies and become the cochlear nerve. Okay, so one more time. Scala vestibuli, or vestibular duct, vestibular membrane, cochlear duct, with a basilar membrane, a tectorial membrane, and the hair cells which make up the spiral organ or organ of Corti. The lower chamber is the tympanic duct or scala tympani, and over here is the spiral ganglion. All right, so that is the cochlea. Now let's go back to this large model and let's talk about the balance centers, the vestibule and the semicircular canal. So this region here is called the vestibule, and it is two enclosed sacs. The upper one is called the utricle, and the bottom one is called the saccule. Inside these two sacs, you will find a sensory organ called a macula, which are hair cells embedded in a slab of jello that has been weighted down with calcium carbonate crystals called otoliths. And so these are the ones that are going to shift in response to gravity when you tilt your head, and it's going to let you know which direction your head is leaning based on which direction the hair cells are bent. Okay, now attached to these two, which are filled with perilymph, is this structure here, which is the endolymphatic duct. And there's usually a diamond-shaped paddle here on the end, which would be called the endolymphatic sac, where we store and then circulate endolymph only inside the membranous labyrinth, okay? So looking at this model, we can also break out bony labyrinth and membranous labyrinth. This ivory color on everything would be the bony labyrinth. And then you notice there's a pink layer that represents the perilymph, the very watery-like fluid. And then the gray represents the membranous labyrinth. The endolymph would be inside the different areas that are gray. Obviously, we don't have them cut apart. We cannot see the endolymph. Okay, so the endolymph is inside the membranous labyrinth. All right, so we did the vestibule, and all of the axons leaving these neurons in here will become this section here, which is the vestibular nerve. Notice we also have pieces coming from these other structures. These structures are called the semicircular canals, and there are three of them for the three planes of direction of movement. The one that sticks up the highest is called the anterior semicircular canal. The one that lies on its side is called the lateral semicircular canal. And the one that is towards the back is the posterior. Now, each of these canals has an area where it connects to the utricle, where it gets a little bit wider. This wide section of the bony labyrinth is called the ampulla. And if we were to look inside, it's wide for the bony labyrinth because it also gets wide for the membranous labyrinth. And inside this part of the membranous labyrinth is a structure called the crista ampullaris, which has a little cupula of jello with hair cells embedded in it. And every time the wave of endolymph comes by, it bends the hair cell inside that organ and tells the brain which direction we are moving and at what speed. And so notice, these are the axons also that are going to come and join the vestibular nerve. Over here, I have a bunch of axons leaving the spiral ganglion, and this is the cochlear nerve. So right now, these two nerves are separate, the vestibular nerve and the cochlear nerve. In some models, they become fused, like this one. Okay. Whoops, sorry. This little one. Showed you the wrong model. This one. And so now we would call it the vestibulocochlear nerve. All right, so those are the details of the inner ear.